So, the ECG or the EKG, it was first time it was recorded by a great scientist whose name is Eintho Wen. So Eintho Wen is the first scientist who recorded the ECG in the human heart. Now what exactly happens in ECG is that there are 3 electrodes, or you can say that leads, electrical leads, which are connected to the human body. So generally a standard ECG machine will be having 3 leads. No doubt there will be like 4 bar, 6 bar, or you can say that 8 or 12. But the most common is 3 leads. So if this is a human body, so the ECG machine it is connected, for example, like this is the first lead electrode, this is the second electrode, and this is the third. So this is the left ankle, this is the left wrist, and this is the right wrist. Now this entire thing makes the heart to be visualized as a closed circuit. So therefore, we are going to understand how this entire thing functions, keeping in mind the depolarization and repolarization which occurs in the human heart. So if you see the human heart, first we will understand how exactly the ECG is initiated. So let us say that it is the human heart. Now here, this is the right side, it is the left side. Towards the right side we have at the corner called as SA node, and here at the junction we are having AV node, sinoatrial node, atrioventricular node, which is present at the junction of atrium and ventricles. Now the electric impulses are always transmitted from SA to AV. Further, AV is going to carry it to the bundle of His, which is running parallelly to the interventricular septum, and then going towards the left branch of Purkinje fibers and to the right branch of Purkinje fibers. So when the SA node is transmitting, as I told you, action potential, so it is going to get the stimuli triggered, it is going to get fired, impulse. Now this impulse is going to start the process of depolarization. So now this SA node, it is nothing but the cluster of cells, mass cluster of cells. So what is going to happen? Throughout this area, the depolarization effect is going to spread from one cell to the next cell, to the third cell, to the fourth cell, to the fifth. Like this, everywhere the transmission or the depolarization impulses, the forces of electrostatic charges are being carried out. So, how it initiates is, it is initially electronegative, as I told you, minus 90 millivolt. Slowly, this impulses is going to make entire atrial chambers electropositive. That is called as a depolarization. So, all the cells will be having the positive charges in itself. So, first the depolarization occurs. Soon the depolarization it covers the entire atrial chamber. It is called as your atrial systole. Atrial systole means contraction, depolarization. Step number 1. Step number 2 is all the depolarized waves will be getting collected into the AV node. And this dotted area, this, like for example, the heart is having the septum in between this. This septum is named as fibrous annula or annulae. It is made up of fibrous annulae. So what is going to happen, all the depolarized signals are going to be collected here. Now you will ask me one question. Madam, why not in this area? Why only at the AV node? Why not here? Because it is made, these fibrous annulae will not be able to capture the electropositivity or depolarized signals. So maximum depolarized signals, when they reach fibrous annulae, it will get damaged or it will be totally become null. And therefore, all the signals will be concentrated towards the AV node only, because it is made up of fibrous tissue, which is not having the capacity to collect the signals, to hold the signals. So therefore, all the signals, so let me draw it very neatly. So the maximum signals will be collected by AV node. Now AV node is made up of, again, it is also a myocardial, complex cells, tightly packed cells. So when these cells are so tightly packed, what is going to happen? What? So when after depolarization and electropositive signals, when they are trying to reach this, because it is a compact mass of cells, very, very compact, the transmission of the electropositive signals across AV node is slow. It is actually very, very slow, because the AV node is not very prominent kind of a structure which can be easily, which takes time. No, it is a very, very small tissue or small mass of cells. So when all the electropositive signals are coming inside AV node, immediately transfer of impulses to bundle of His is not very, very quick. It is very slow. One more scientific fact is that at the AV node, when all the depolarized effect or the signals are coming, it is the junction between atrium and ventricle, right? So when the signals have a depolarization effect have to cross atrial to ventricle, it is kind of a holding point. It's not like suddenly atrial to ventricle. No, there is a pause. So this AV node helps in collecting entire signal, hold it. It's a kind of intermediate station, junction, where the electropositive charges are held for a while, for just few fraction of seconds. That is why the conduction is also very, very slow. Depolarization occurs here. Positive charges are moving here, no doubt about it. But it is taking at a very, very slow pace. One and third reason. It also, while holding, when it holds the charges together, electropositive charges, it also gives atrial chambers time. It gives time to the atrial chambers to contract properly, so that atrial systole is effective. It is not very rapid. It is very, very. But compared to AV node, the atrial systole or contraction or depolarization is little faster. Not very fast, but it is faster. And if you can see that from SA node, all the depolarization signals are passing which side? Two sides. One is downward, it is downward like this, and towards the left side. Yes, sir. Towards the left side, because it is located like this. So downward and left side. So this direction is also very important when we are making the ECG graph. Next one. This AV node, it will transmit all the depolarized signal to the bundle of His. Now bundle of His, very, very important. This bundle of His, if you see, if you take a piece of interventricular or this bundle of His only, you will see like this. Cells are there, very tight, again tightly packed cells. In between this tightly packed cells, in between, there is a free space or the channels. There are some large cells also, and these cells will be having what? Lot of gap junctions, multiple gap junctions. So compared to AV node, the bundle of His myocardial cells, the electric impulses will be faster. It will travel very fast. Multiple gap junctions, more area, the electric impulse will not take much time. Aram se easily it will come out. So here at the bundle of His or Purkinje fibers, the depolarization effect is very, very fast. See here, the depolarization from AV node travels like this for a short period of time. It is a very short period of time. Now it will go like this, it will again go like this, interventricular septum. From the interventricular septum, it will spread to the ventricular wall here. Then the depolarization will go to the like this. So this is how the electric impulses are being traveled. And once when the depolarization effect is completed here, now next is there is a repolarization of the ventricles. It means what? When entire ventricles are getting contracted, everywhere there is plus charge. Here the plus charge is actually ending like this. And when the plus charge is ending, what is going to happen? Here, the repolarization effect is going to, it means what? It is going to become relaxed. It relaxes. So, it is followed by atrial systole, then is ventricular depolarization, After ventricular depolarization, ventricular repolarization. So, step 1, step 2, step 3. All these steps are going to be plotted on the ECG graph. So, let me show you how it looks like.